Great. Okay, we can probably just about get started. And I'm very, very excited to introduce Professor Olaf Sporns today. He is the Provost Professor at Indiana University in Bloomington and is one of the most influential thinkers in our field, it's fair to say. His work has helped shape the very framework through which many of us study the brain and the brain's organization. And he coined the term connectome, which many of us use several times a day, and has produced some incredible work revealing how this connectome gives rise to the brain's functions and functional capacities in health and in disease. Professor Sporns has been a driving force behind the field of network neuroscience, not just through research, but also through his leadership as the founding editor of the journal Network Neuroscience, and also through some landmark books like the Networks of the Brain and Discovering the Human Connectome. His ideas really have inspired countless researchers across disciplines, I think, and I'm sure will inspire several more on this call today. So yeah, it's a great pleasure to have you, and let's get going.
Thanks so much, Petra, and great to be back after a few years in this series of talks. And as you mentioned, Petra, my work is focused on network architecture, complex systems related to the brain. So I want to give you a perspective on that. I will touch on a few old, greatest hits, but also on a few new studies that we've produced the last few years. So hopefully they'll be interesting to the audience. Um, I don't need to explain to you how important brain networks have become in our field, but I do remember a time 25, 30 years ago when networks, at least in the sense that we use the term today, have not been mainstream neuroscience at all. Now they are found in really all sorts of different systems, from invertebrates to rodent brain to the non-human primate, and of course to the human brain, across both structure and function. An enormous amount of work has been carried out in the last 10, 20 years. And I want to give you a few of the highlights from my own perspective. Hang on, what is happening? I need to get back into my presentation. Have you lost?
Sorry, I was muted. Now we're seeing, yeah, we're in. We're
Okay,
okay.
we're back in.
Yep.
Okay, hopefully that won't happen again. So at first, a few words about connectomics and architecture, mostly structural connectivity, and then we turn to dynamics and then information theory at the end. The dichotomy between structural and functional connectivity is fundamental in our field, and so I want to very briefly highlight this dichotomy, illustrating it in the example of the human brain. The anatomy of the human brain has become accessible to us in a non-invasive fashion through diffusion imaging and tractography over the last 20 years. And my colleague Patrick Hagman and I started building network representations from these data a number of years back, which we then also correlate with resting state brain dynamics, an emerging field that has really taken over cognitive neuroscience by storm. These two representations, a structural network on the left and a functional network on the right, are mathematically quite different from each other. The structural network, typically sparse and amenable to graph analysis, very straightforwardly, I would say. Functional networks often derived from time series data, similarity matrices or correlation matrices in the case of fMRI. And I would want to stress that it's mathematically a very different object. All pairwise relationships exist, for example. It's not sparse that way. And I would say there's a different set of methodologies that we want to apply to the analysis of functional networks compared to structural networks. When we first started looking at the human connectome with a grand total of 5 subjects in our first study, something which would not be publishable today, I suspect, we found some fairly big features that have, I think, held up very well through many, many replications. Each cortical area has a unique regional fingerprint. There's a broader, perhaps heavy-tailed degree distribution, with many regions having few connections and a few of them having quite a few more connections. There's high clustering and short path length, the two hallmarks of the small world. And there's prevalent modular organizations, modules in the brain interlinked by heavily connected hub regions, which in turn are also connected to each other more densely than expected by chance into a structural core, or sometimes we now call it a rich club. These features have held up really well across many, many applications and have display significant individual variation and vary with across participant groups, also with clinical status. All of this has been a very rich field in the last 15, 20 years for analysis and modeling. The connectomics obviously has also been carried out in model organisms, all the way from invertebrates to rodent brains and non-human primates. And this summary slide is just trying to illustrate, first of all, that very many different methodologies for acquiring structural connectivity data can be employed, from optical imaging to track tracing to literature collation. And secondly, that when we apply these methods and we construct connectome matrices for different species, we often find a common set of features, such as the existence of modules and the existence of hub regions which are interconnected into rich clubs. This gives rise to a question, why are these features so commonly observed and so conserved across so many different related animal species? I want to mention very briefly that we're now venturing into the realm of EM reconstruction data sets with a vengeance. The adult Drosophila connectome was published last year, quite large data set that's already been studied with graph theory tools by a number of groups. And so I think we have a very rich future ahead of us with lots of data and with lots of opportunities for analysis and for modeling. Here's where I want to highlight a study with my old friend and colleague, Ed Bullmore, a review article we wrote, I hate to say, a dozen years ago now, where we asked the question why those common features might arise. And in biology, when you ask a question about why something looks the way it does, you often immediately come into the realm of evolution. And you look for factors or design features that have served as constraints for shaping the architecture the way it looks today. And so our proposal at the time, our framework, was that there's really a tug of war or a trade-off going on between two different tendencies. On the one side, the brain wants to be cheap. Wiring minimization is a principle that's been around in neuroscience for over 100 years. But if we optimize the brain for wiring length or minimize wiring length exclusively, we end up with a brain that is, looks like the brain on the left, really not optimal for moving information from one end to the other. On the other hand, if we want to optimize information flow and want to keep path length extremely short, for example, we end up with a random topology, which in turn is not at all minimizing cost. So there are these two opposing tendencies, one in terms of minimization of cost, the other one maximization of efficiency or information transfer. These two trade off against each other, and what we end up with is a compromise, if you wish, a trade-off that, for example, preserves cost in the context of modules which are internally dense and locally clustered, and then long-distance connections between them, which optimize information transfer. That was our proposal, our framework. And for a number of years, we weren't really able to test this framework very well because data was sparse and very few animal species had been imaged or had been reconstructed. However, more recently, we got together with colleagues in Tel Aviv University, Yaniv Asaf and Yossi Yovel, who had accumulated a very interesting database of mammalian data sets from brains of a variety of species, over 100 million species, collected in the wild or in zoological gardens, and then imaged post-mortem and reconstructed with tractography. The connection matrices, a few examples of them are shown on the left. What you see on the right is what we call a co-classification matrix, which is the propensity for two pairs of nodes to be affiliated with within the same module. We are applying modularity optimization to multiple scales, then construct a so-called agreement matrix, or here called co-classification matrix, which then defines for each pair of nodes how often this pair is found in the same module. It's sort of like a distance metric or similarity metric that quantifies affiliation within the same modular arrangement. A very useful construct, by the way, for all sorts of analyses, including human MRI data. Here it's applied in the context of the mammalian dataset. So we set out to explore whether we could compare these different connectomes from different species to each other and what patterns we might find. My grad student at the time, Josh Faskowitz, made the first step. He retrieved genetic data for all these species from an online database and constructed a phylogenetic tree, which tells us something about the distance between different species in terms of their genetic variation. Secondly, he then asked if we look for more closely related, phylogenetically related species, are there connectomes more similar to each other than for species that are more distantly related? That is sort of like a gut check or a validation check. We should be finding that if our data is gives us what we're looking for. So we were very interested in that first result, and indeed it holds up. So more closely related species within the same taxonomic order in the mammalian system have more similar connectome architectures than more distantly related species. The second point relates to brain volume. It immediately occurred to us that mammalian species have brains that range in volume over 3 orders of magnitude. The smallest brains from the smallest bats are only 1/1000th the volume of the largest brains in the sample from primates and some rodents and some hoofed animals and some carnivores. Brain volume plays a big role, as we all know, in allometric relationships between gray and white matter and in evolutionary relations as well. So we wanted to see whether, for example, the larger brains showed different relation to geometric space than the smaller brains. For that, we took off the shelf a generative model that fixes the locations of brain regions and then assigns connections based on geometric distance between them using an exponentially decaying probability rule. The magnitude of the exponential parameter, how big it is, and it's typically negative, tells us something about the severity of the penalty that long distance connections are subject to. As we see, as Josh did for all these different species, if we fit that generative model to the geometric distribution of brain regions, we find that the smaller brains incur a smaller distance penalty, a smaller magnitude of this parameter, while the larger brains are much more penalized for long distance connections. In other words, long distance connections in larger brains are much less frequent and subject to a much more severe distance penalty. That was our first inkling that brain volume plays an important role in addition to phylogeny in terms of defining connectome architectures and differences between them across different species. Secondly, my former postdoc, Maria Grazia Puchedu, took this line of work several steps forward. A nice paper published last year in PLOS Biology. I'm just giving you the gist of this. We looked systematically at the relation of anatomical modularity to brain volume across these over 100 different species. And just to give you one little snippet of the data that we came up with, when we're looking at the propensity of two brain regions of being in the same module, which is our co-classification value, or CC, and we look at the weight of a connection between them, if it does exist, we find that the relationship strengthens as the brain volume increases. In stronger brains, the existence of a connection between two pairs of brain regions is more strongly related to their propensity of being within the same anatomical module. An inverse relationship ensues for Euclidean distance. Overall, the study gave us the following sort of summary account. As brain volume increases, modules become internally more dense, spatially more compact, that is, brain regions are found in the same volume of space, closer to each other, and they are less interconnected with each other. This reflects the distance penalty I mentioned earlier. So we're getting to in larger brains to an organization that's much more modular, spatially organized, and then sparse with respect to interconnections between the modules, placing a very high premium on their existence and on the ability of these connections to link signals between different modules to each other. So brain volume and brain architecture are interrelated, and modularity plays an important role in how this relationship plays out. I want to briefly mention another line of work I'm pursuing with Larry Swanson and Joel Hahn at USC, looking systematically at different subdivisions of the rat brain, not just the cerebral cortex, which we started with, but also very many other subdivisions that we are often neglecting in our human fMRI work, where it's often the cortex that is the most important thing that we pay attention to. Just as a survey on the lower left, the brain can be divided into various subdivisions, as you can see here. The cortex in the rat consisting of 154 regions, but the brain as a whole consisting of almost 800 brain regions with about 72,000 connections between them. This data is derived from systematic literature survey of existent literature on track tracing in the rat brain, which is abundant, and which Larry and Joel have systematically collected and archived. So we have a connection matrix that's directed, weighted, quite sparse, and comprises a very large fraction of the complete rat brain. Several messages from that work that I want to convey to you here very briefly. One is that in the context of this work, we worked out and then optimized an approach to modularity, which is based on hierarchy. Modules are basically partitions of nodes into subsets that are internally densely connected, usually in associative modularity. But it turns out that if you look at multiple spatial scales, you can come up with partitions of different grain, if you wish, of different level of detail. It's very important to me to get across to you the point that we should not view modular partitions as being unique and uniquely defining a particular brain architecture, but there's actually valid partitions at different levels of scale that are nested within each other. So it's a set of modules within modules within modules. We can access that by clustering the co-classification matrix, essentially, that I mentioned to you earlier as a new construct. And in that way, we can get a much more complete picture of how modularity plays out in a given brain architecture. This applies, by the way, also for human fMRI data. We've used this approach in imaging data sets as well, but here it's applied in an anatomical data set from the rat. So that's message number one. Modules are partitions. These partitions are hierarchically nested. And if by looking at the entire hierarchy of this nested architecture, we get a much more complete picture of how modularity plays out in a given brain. Secondly, brain architectures, the types of sort of global brain architectures that we might find, for example, small worldness, plays out very differently in different subdivisions of the brain. I want to really underscore that because to me it stood out as a something I didn't really expect to see. The larger subdivisions of the brain as a whole, the central nervous system, the forebrain, the end brain, the rostral sector, the cerebral cortex, yes, those have hallmarks of small world organization in the sense that there's high clustering and a short path length. However, other brain subdivisions, the cerebellum, the thalamus, the hypothalamus, the myelencephalon, the spinal cord, do not express these properties at all. And so there's a great heterogeneity of architectures that we come across when we look outside of the cerebral cortex and look at other subdivisions of the brain. This is something that intrigues me because, for a while, it looked as if every brain had small world architecture, so what's the big point? It turns out that if we look more closely in more detail, different subdivisions of a mammalian brain, such as the rat, may have actually very different architectures that probably express their very unique and specialized information processing requirements. So much for universality. It's actually not as universal as we might have seen early on. Now let's turn over to dynamics. Of course, the brain is active over time, and as we all know, resting state functional connectivity has become a very widely used tool in our field. And in our lab, a core question has been, how do we get from a static anatomical connectivity pattern, such as the one on the left, to something which is as dynamic and fluctuating and ever changing as the ongoing dynamics in the human brain shown on the right. As we all know, we construct resting state networks by taking individual frames of activity, snapshots of activity in the brain, cross-correlate them, clustering those cross-correlation matrices, which then gives us these blocks that we see in this diagram on the main diagonal of internally cohesive sets of regions that are more or less expressing similar activity patterns over time, and that we now have canonically named, such as control system, default mode system, dorsal attention system, and so forth. This has become the lingua franca really in cognitive neuroscience, and I can't underscore how important a shift that's been, because 20 years ago, many of these systems did not exist, at least not in the way that we refer to them today. And at the bottom here, just a brief shout out to some of the canonical studies carried out a while ago, which have shown us that despite the fact that resting state is so underspecified in terms of the experimental paradigm, it gives us something very reproducible even from different subject cohorts and different imaging centers. So there's something very reproducible, very concrete about these systems that we've now been familiar with for a number of years in our field. But I would argue that functional connectivity itself is dynamic. It changes over time. And, I've written about this in my own sort of more theoretical musings in the past, proposed that functional segregation, functional integration are probably waxing and waning, interacting with each other over time and the brain going at one point in one direction or the other. Ideas that I think have found some experimental support, including in work from Andrew Zaleski and Michael Breakspear and Russ Poldrack and Max Schein, just to name a few of the examples of early work in this area. I could cite many other authors who have since shown that functional connectivity in rest and in task is dynamic and fluctuating. We have, of course, pursued that line of work in our own lab as well, working with my former postdoc, Makoto Fukushima. What he did is he looked for episodes, windows of functional connectivity, small, short episodes of about 30 to 60 seconds, where modularity was high or low, higher or lower than expected. These so-called maximum and minimum Q episodes had particular functional connectivity signatures to them. As you can see at the bottom of this diagram in two different data sets, what you're basically seeing is default mode and control regions sort of breaking off and being anti-correlated to those regions that are more related to sensory motor function or attention. In a sense, the external and the internal part of the brain co-fluctuating against each other. This is a hallmark of these high Q or high segregation, high modularity states, while the more integrated minimum Q states are much more flat in terms of the shape of the correlation matrix that you see at the bottom. So we pushed this line of work doing windowing on resting state and task for a number of years, but quite frankly, we were not satisfied with the approach to some extent. Lots of assumptions go into the construction of these windowed representations. Modeling assumptions have to be made as to how we distinguish different states from each other. And there are many free parameters that I felt very uncomfortable over time choosing. So a period of creative disorganization ensued in the lab. Took a picture of our blackboard right before the pandemic essentially put a halt to all this. And it's a sign of of creativity when new ideas come out, right? So there's actually at the bottom of this bottom right-hand corner, there's an idea that emerged from one of these whiteboard discussions that I'm going to now expound a little bit in the next few minutes. And that is the idea that we can actually unravel the classic functional connectivity into moment-by-moment contributions. And this idea really was brought to the fore by Josh, my former grad student, and Rick Betzel, my former colleague here at IU now at Minnesota. The idea is very simple, actually. What is cost correlation really? One of the mathematical definitions of it is that it's the mean of the element, the moment-by-moment product of the two time series. So let's look at the bottom. We got two bold signals, region I and region J, varying over time. So that's familiar to us. If we now multiply these two nodal time series together moment by moment, we are getting what we call an edge time series, which is the co-fluctuation of the edge ij over time. If that signal is positive, the two regions, the two nodes are either both positive or both negative in terms of their bold activity. If the signal is negative, then they disagree with each other. Think of it as an agreement or co-fluctuation of the pair of nodes over time. Now, the neat fact is that when we take the mean of the edge time series displayed here, it's exactly equal to the functional connectivity. So functional connectivity is a construct we're all familiar with. We now unravel it, and we can look at the moment by moment single TR contributions for each node pair. Here's what this looks like if we display it in a matrix form. We have edges on the y-axis and time on the x-axis. If we take this matrix and we take the mean of that matrix along the y dimension, we're getting a vector, which when we fold it back into square form, is exactly equal to functional connectivity. So what's on the left is familiar to us. What's on the right was perhaps somewhat new at the time. Now you notice these vertical streaks, which gave us a bit of a headache early on because we were worried about fMRI artifacts kicking in at certain moments in the scan. These moments of high co-fluctuation, we call them events initially, and they stand out from the rest of the distribution because at that moment in time, co-fluctuation patterns in the brain have very high amplitude on average. These events occur in really all resting state or movie or task scans that we've ever looked at from different imaging centers and from different subject cohorts. These are intermittent episodes of high co-fluctuations, these events, and they are brief and somewhat stereotypic, actually. Quite a lot of work has been done, especially in Rick Betzel's lab, looking at the spatial and temporal distribution of these events and what they tell us about underlying functional interactions. I just want to point out here that all of the moments in the edge time matrix contribute to static FC because they all go into the mean that ultimately gives us FC on the left. But some moments, for example, these high co-fluctuation events, contribute somewhat more than the others. Okay. Now, as part of my pandemic project, when I was stuck at home like everybody else and had nothing to do but work, I tried to develop this methodology a little bit further, and I made the following little movie that I want to show you. And I show this in all of my talks because I think it builds intuition as to how we might think about this. On the left is a functional connectivity matrix, which is actually derived from the single resting state scan that I'm going to show you in just a minute, coming from the Human Connectome project. So that is a construct on the upper left we are all familiar with, and I think we have all agreed is useful and meaningful in our daily work. When I start the movie, you'll see that the edge time series are starting to populate. So edges on the y-axis, time on the x-axis. You'll see the amplitude on the bottom. There was just a very high co-fluctuation event occurring. What I want to direct your attention to is the moment-by-moment pattern of co-fluctuations displayed in this matrix at the top. Essentially, the mean of all the frames, all the movie frames that you're seeing in this little movie is exactly equal to what we see on the left. But I would like to stress that at no point do you actually see the pattern on the left appear in this cofluctuation matrix. Red color means positive cofluctuation, blue color means anti-cofluctuation or negative cofluctuation. The familiar architecture, the functional systems that we see on the left are rarely expressed uniquely and jointly in this momentary cofluctuation matrix. That's an important point, I think, and that's the reason why I'm showing this to you, because it drives home the observation that these patterns of functional connectivity that we see on the left really are superpositions of many, many elementary states that occur at much, much faster timescales. I will sidestep the popular discussion as to whether or not functional connectivity is itself stationary or what's going on there with dynamics. We don't need to do that because all we do is we take what we already know, functional connectivity, and we decompose it into single moment contributions. We can take this approach in many different directions. A couple of things I want to point out and emphasize here to you. At the top is our edge time series matrix. We've already seen that before. We can reconstruct functional activity from that in a very straightforward way. At the bottom is a binarized version of this exact same data where we've thrown away the amplitude information completely. We've thrown away all of the bold amplitudes. We just retain the sign. Are bold signals above or below their respective mean? It turns out that this is a binary pattern, first of all, a pattern of zeros and ones, very highly compressed from the original time series. Lots of information has been lost. And yet, if we build an agreement matrix, a co-classification matrix from that construct, we are getting back something that is stunningly similar. Actually, correlation is 0.96 or 0.97 to the original functional connectivity. It's a very different way of building, it's a somewhat different way of building a functional connectivity matrix based on a binary signal that's been retained, and yet very, very highly preserves what we see in the original data. That is I think an interesting observation that could be leveraged in many different ways, and we have done so in our lab in ways that unfortunately, I have no time to go into in much detail. Second point I want to make is underscore that the brain functional systems that we're so familiar with, visual systems, motor, dorsal attention, you name it, are not continuously expressed, but in fact only emerge if we superimpose many, many of these individual moments of cofluctuation on top of each other. If we look at whether cofluctuations occur within these systems and we apply an appropriate null model, we actually see that these systems are not constantly expressed, but only intermittently, and in fact in different combinations with each other. A very rich analysis path that I think has not been pursued very much, also not in our lab for lack of time. But I wanted to mention it here. We can actually build without making any free parameters really, or making assumptions about hidden Markov models or k-means clustering or who knows what. We can build state and state transition probability matrices very handily from such an approach. Finally, work using a single TR decomposed fMRI has been taken forward by my former grad student Sarah Katz now at Yale for doing fingerprinting and showing that brain behavior relations as well as fingerprinting is actually temporally expressed in short moments and can be optimized to give better signals if we choose the right moments using objectively defined criteria. A couple more points about this, and then I'll move on. Work that was largely carried out in Vic Betzel's lab has shown that movie data, actually, in movie data, we can see that events or high amplitude cofluctuations occur in moments that are synchronized across individual subjects. This suggests that these are not these moments are to some extent stimulus driven or stimulus locked, and work by Farmers as Falani and Jake Tanner has dug into that aspect a great deal more. And finally, a recent collaboration I was able to carry out with Tyler Santander and Mike Miller at at UCSB and colleagues in Germany on individuals who have undergone callosotomy, some of them partial callosotomy as shown in the top row, and some of them complete callosotomy at the bottom. These are epilepsy patients who underwent the surgery in the context of their clinical regimen. What we see here is that when we look at, first of all, the tractogram confirms that the callosotomy was partial in the top case and complete in the bottom case. If we look at edge time series for the two hemispheres separately, left hemisphere and right hemisphere, in the case of a patient with partial callosotomy, where some of the callosal fibers have been spared, we continue to see fairly strongly highly correlated amplitude co-fluctuations of edge time series across the two hemispheres. But in a patient where the callosotomy is complete, so the two hemispheres have been totally disconnected from each other, at least through the callosal fibers that have been cut, the edge time series co-fluctuations have now become largely desynchronized. This is a strong suggestion, I would say, that the synchrony that we find in these co-fluctuation patterns is not some measurement artifact or noisy manifestation of a stochastic process, what have you, but rather reflects a physical origin of these co-fluctuations in structural connectivity, because that is what's been manipulated in the callosotomy. And consistent with that idea and preceding this experimental finding by a few years, actually, my former grad student, Maria Pope's work in implementing neural mass models or simple phase oscillators like the Komodo model, and then building generative models of functional connectivity where we can actually look at whether or not these fluctuations occur. It turns out quite stunningly, in my view, that these edge time series co-fluctuations, co-fluctuation patterns, including events, are reproducing very nicely without any additional model assumptions or free parameters in our simulations. This is an example of an edge time series plot with underlying amplitude co-fluctuations from such a model, and without making any additional, without tuning anything about the model itself, really, we're getting the same kind of phenomenology that we get in the empirical data as well, without imposing any mechanism on the model that drives these co-fluctuations internally. So where are they coming from, you might say? They are depending on the modularity of the underlying structural connectivity matrix. If we give the model a modular SC matrix as its coupling structure, we then see moments of high co-fluctuation occur. This is no surprise to people who come from a physics background, because the phenomenon of cluster synchronization has actually been well known in synchronization models in physics for decades. Models that lack structural connectivity modules will not show these high co-fluctuation events occurring. So we have, at least from the modeling side, additional evidence that these fluctuations that we see when we unravel functional connectivity into single DR contributions actually have causal mechanistic origin that have to do with the interplay of dynamics and connectivity. A brief mention of a whole line of work that I'm very fond of as well, somewhat separate from what I've talked about previously, and that is to do with network communication. In a sparsely connected graph, such as the human brain, for example, if a message is to be passed between two nodes, usually it does not, there's no direct connection between them. That's the definition of sparsity. So a signal would have to travel across a multi-step path between node I and node J, if I is the source and J is the target. Now, finding these paths that are short and perhaps even shortest is a computationally hard task to solve even today. And how would the brain know which path to use if a signal is to travel across multiple steps? This is the communication problem that I think is looming large when you have a sparsely connected network. One way to address it is to take connectome data, connectivity data coming from empirical, let's say, diffusion imaging and tractography in humans, implement a communication model on top, such as diffusion or shortest path signaling, which then gives us a communication matrix, which is a prediction as to, under the policy, how easy it is or how hard it is to pass signals from node I to node J. This communication matrix can then be compared to empirically measured constructs such as functional connectivity, which perhaps intuitively might carry the same information. How hard is it to get from A to B in a network? If the correlation is high, if functional connectivity is high, it might be quite easy. This work was started in my lab by Joaquin Goni, my former postdoc, now faculty at Purdue. We tested a variety of communication strategies and policies, one of which stood out to us, which is called search information. And that is, given a shortest path from a source to a target, how many decisions have to be made walking along that path to get to indeed discover that the target is on the shortest path. The number of decisions relates to the degree of the intervening nodes between source and target. The higher the degree, the more off-ramps there are, the more ways to get lost, if you wish, the more hidden the shortest path, and the weaker we would predict the functional connectivity. It turns out that that idea fits remarkably well with empirical functional connectivity. The search information scales very nicely, shows a high correlation with empirical functional connectivity, and shown on the right in this fancy diamond plot. On the left, an empirical functional connectivity matrix, a cross correlation of fMRI signals from a single brain. On the right, a predicted signal based on that brain's anatomical connections and search information, plus a few other communication policies combined. So a very, I would say, significant prediction capability. And the nice thing about this model is that it's actually analytic, so you can actually run it in MATLAB in under a second. The work was carried forward by my more recent postdoc, Kyle Seguin, and he tried to address the problem of different communication policies empirically, leveraging a data set coming from a clinical set of studies where patients had been implanted with depth electrodes in their cerebral cortex, again, pre-surgically in preparation for epilepsy surgery. These electrodes allowed physiologists to inject current in one electrode, for example, and then measure responses in the other electrodes. If we now overlay this setup, the electrode locations, with a structural connectome, we can now relate the essentially effective response at target locations with the length of paths and different communication protocols potentially that link source to target. So doing this and implementing different communication models on top of a human connectome, that gives us a prediction of how easy it should be or how hard it would be to move information from a source to a target. This can then be related empirically to the empirical data. And Kaio did that systematically for a number of different communication protocols, including search information, abbreviated here as SI, which emerged as the winner in this particular contest. So once again, search information being in the lead among different communication protocols. This is a way in which we can test whether or not multi-step paths are in fact used and how they are used in brain communication processes. Finally, Kaio summarized a number of different ideas and studies and communication models in a review article. And just to give you a brief overview here, we've got a spectrum of different communication strategies, from random walks, which are like diffusion, to shortest paths, which are often seen in technological applications, but actually hard to implement in biology. And they have different requirements in terms of the information they require, different energy requirements, different efficiencies associated with them. And I would say it's an open question where on the spectrum brain networks lie, and in fact, whether perhaps we're using a combination of these strategies in communicating in the brain. So that is a whole line of work that I think is very fruitful and is being carried forward by a number of people, including Kaio in his most recent work. Okay, finally, a few words about information and information theory. And here I actually connect back to something that I have worked on in my career really more than 30 years ago with Julia Chinoni and Gerald Edelman back in the old days. Recently, there's been a resurgence of interest in functional integration that we can characterize and measure using different measures of multivariate information, which is really another way of saying how can we leverage not bivariate but higher order dependencies in neural time series to express how different signals relate to each other and perhaps integrate information. Now, there's two different ways to think about information integration, and I hope the following analogy is not too simple-minded to you. On the one side, think of the string section in an orchestra where everybody in the string section more or less plays the same tune, at least at some points in the composition. That's kind of an example of redundancy. So we're duplicating identical information. If we lose one of the string players, not much is lost in the overall picture. And there's what we call, in information theoretic terms, high total correlation. There's high overlap in terms of the information content. The information of what is the gist of what is happening is disclosed by looking at every single string player separately. In fact, every single string player, if they play the same tune, is identical to any other. However, in a string quartet, each player plays their own line. And looking at an individual player and taking the other players out of the composition makes no sense at all. The composition is lost completely. The composition is only disclosed if we look at the joint state of all the players at the same time. That's an example of synergy, the integration of unique information over time. And in information theoretic terms, it's often now associated with what we call negative O information, which is a particular information theoretic metric we can calculate from time series data. So the question we might ask is, do both redundancy and synergy exist in the brain? And if so, when do they exist and where are they expressed? And here I'd like to really give a big shout out to lots of people in the field, some of their names given below, whose work I recommend to you if you're interested in this line of work. And it's a very exciting and emerging area, higher order dependencies and synergy and redundancy in complex systems in general, but in the brain in particular. In our own work, we've tackled both redundancy and synergy. First, dealing with synergy, work by my former postdoc, Maria Grazia Puchedu, we've argued that functional systems, such as visual, some motor, default mode, and so forth, classical systems that we've worked with in fMRI, in the fMRI space for many years now, really are expressions of redundancy, internally redundant sets of nodes that more or less do the same thing over time, are highly correlated with each other. We hypothesize that we can measure this internal redundancy of these functional systems using the information metric of total correlation, or TC. And as you can perhaps see from this formalism, it is basically a multivariate extension of the bivariate mutual information. And we can calculate it under Gaussian assumptions analytically from the covariance matrix. When we do that, we can then construct a cost function by finding a partition in our network that optimizes the total correlation score for a given set of brain regions and given sets of brain regions that form a partition of the system as a whole. When we do that, and Maria wrote a whole software tool which is available publicly on GitHub to do this well, we find optimized partitions that optimize this information theoretic quantity. So this is very different from Q maximization, that are looking quite similar, although in characteristic ways also different, to canonical systems coming from other types of analyses, such as the ones proposed by Thomas Yao many years ago. In her work, Maria also made comparisons between these systematic comparisons between these different approaches and looked at the development of these informationally optimized partitions across the lifespan and in different data sets. It's a very different way of, I think, very different way of defining partitions, not based on Q maximization, but based on the maximization of an information theoretic cost function shown on the left. Lastly, synergy, pushed forward by Maria Pope and Thomas Varley, two of my former grad students, is related, interestingly, to an very old measure of complexity that Tononi and Edelman and myself came up with, now called TSE complexity, and relates total correlation and what's called dual total correlation to each other, and negative O information results when the difference between these two quantities becomes negative. We asked the question, being able to calculate this, for what parts of the brain do we find that synergy outweighs redundancy? In other words, the O information is negative and the subsets that we find are synergistically dominant. We find that these synergistic subsets are actually interspersed between classical functional systems. So they don't correspond to one functional system, default mode or visual or smarter motor, but instead sit at the interstices of these systems, combining voices or nodal time series from these individual systems together. When those nodal time series become integrated, think string quartet, a composition emerges, which is synergistic in the sense that the information is contained only in the joint set of the information and not in the individual nodes themselves. The nodes themselves come from different communities. So synergy is found between classical functional systems and is expressed in human fMRI data. Maria Pope also published more recently an account of when these systems are highly expressed in time, something we can tap into using essentially like a decomposition of information theoretic metrics such as O information over single TRs. Finally, the global picture here is one where networks on one side interact with dynamics on the other. Much of the talk today has covered different aspects of network organization, connectomics, obviously, modularity, aspects of cost, things like that, and then also synchrony, communication patterns, and connectivity dynamics under the rubric of dynamics itself. Interaction between these two then gives rise to information and fluctuating patterns of synergy and redundancy, higher order interactions, and ultimately complexity. So in my career, I've sort of tried to triangulate between these three different aspects of these three different perspectives that we can use to look at brain function globally. And while I've spent a lot of my time on the side of networks, more recently especially, I would like to emphasize that it is important to understand the interaction of networks with dynamics on top, and then the information, theoretic quantities and structures that emerge from that. And I think there's some much work to be done in the intersection of these three different dimensions. With that, I'll stop. I'll mention that Network Neuroscience is a journal in the field that I started 10 years ago. I'm now about to step off as editor-in-chief, and Alex Fornito will take over at the end of the year. I recommend the journal to you if you have relevant work in this area. I have many people to thank, and with that, I stop. Thank you very much.